Welcome students. Today we are going to discuss mutation and its types. Mutation is the ultimate source of all genetic variations and is the raw material for evolution. Had there been no mutation, all the genes would have been present in only one form and there would have been no evolution and no adaptation to the environment. On the other hand, too frequent mutations will disturb the entire process of the transfer of genetic information from one generation to another and would be biologically unacceptable. Mutation can occur at any stage during the development. For example, number 1, if mutation occurs in a germinal cell before differentiation of gametes, it would influence several gametes and will thus influence all the individuals derived from these affected gametes. Number 2, if mutation occurs in a gamete or a zygote, a single individual will carry the mutation. Number 3, if mutation occurs in a cell after the zygote has undergone one or more divisions, only a part of the body will show the mutant character. Such mutation will be called somatic mutation, in contrast to germinal mutation. Various classifications of mutations are known, each based on a definite criteria. We will discuss them one by one. Number 1, mutation based on direction or forward mutation, reverse mutation. Now, what is forward mutation? The change from wild type to mutant form is called a forward mutation. For example, the wild type long pea plant got mutated to dwarf plant, and this dwarf plant does not reverts back to long pea plants. Number 2, reverse mutation. If a mutant is again mutated at the same site in such a manner that its phenotype returns back to the wild type, the mutation will be reversion of the first mutation or a back mutation. The back mutation can be of two types. Number 1, true back mutation. 2, suppression mutation. True back mutation, where the second mutation is at the same genetic site where the first mutation had taken place, and the phenotype as well as the genotype of the organism revert back to the wild type. 2, suppression mutation. In such mutation, the second mutation is at a site other than the site of the first mutation. However, the second mutation nullifies the effect of the first mutation, and the phenotype of the organism is back to the wild type. But the genotype of the revertant will show the mutation at two separate locations. Number 2, mutation based on the cause, spontaneous mutation and induced mutation. Spontaneous mutation. In this type of mutation, there is no apparent reason for the change in the DNA. These take place spontaneously, without any known cause. The deamination of bases, especially of cytosine, and the spontaneous depurination are good examples of this type of mutation. Induced mutation. In this type of mutation, change is caused by certain physical or chemical agents. These agents are known as mutagens, and exposure to these mutagens results in mutation. Mutation can be induced through two types. Number 1, a radiation-induced mutation. Number 2, chemical-induced mutation. Now, what is radiation-induced mutation? A number of radiations, especially the electromagnetic waves with 100 nanometer or smaller wavelength, cause ionization. The example are the X-rays, gamma rays, and cosmic rays. Effect of ultraviolet radiations. Ultraviolet is a relatively weak mutagen. The normal level of UV in the sun light are not strong enough to cause mutation. Any damage to the DNA is repaired readily by the cell. UV is absorbed by both the purine and the pyrimidine bases, and these are converted into an excitable state which are more reactive. The UV radiation of 254 nanometer or lower wavelength is more damaging. It can cause the formation of pyrimidine dimers. Number 2, chemical induced mutation. A number of chemicals are mutagenic in nature and are grouped into two classes. Number 1, chemicals that can cause mutation to both the replicating and non-replicating DNA. Number 2, some of the examples of this class of chemicals are nitrous acid, alkylating agents, etc. Number 2, chemicals which can cause mutation to only replicating DNA, such as acridine dyes and base analogues. Now, what are these mutagens? These are, number 1, nitrous acid. It causes oxidative deamination of the base in DNA. Number 2, alkylating agents and hydroxylating agents. These chemicals transfer a methyl or ethyl group to the bases such that their base pairing potentials are altered and transitions result. Number 3, acridines. Acridine dyes cause the frame shift mutation. Number 4, base analogues. A number of modified bases can be incorporated in place of a normal base during the DNA replication, as the DNA polymerase cannot differentiate between the normal base and its analog. Mutations based on the tissue of origin are somatic mutation, germinal mutation. Now, what is somatic mutation? Mutation in somatic cells are called somatic mutation. The mutation is not passed along the next generation by sexual means. To maintain this mutation, the individual containing the mutation must be cloned. Two examples of somatic clones are navel oranges and red delicious apples. Horticulturists first observed the mutants. They then grafted mutant branches onto the stalks of normal trees. After that, the graft was established. Cutting from the original graft was grafted onto tree stalks. In this way, the mutation was maintained and proliferated. Now, germinal mutation. If a gene is altered in a germ cell, the mutation is termed a germinal mutation because germ cells give rise to gametes. Thus, the mutation is passed along the next generation by sexual means. Typically, germinal mutations are not expressed in the individual containing the mutation. Mutations based on the type of trait affected are morphological or visible mutations, biochemical mutation. Now, what is morphological mutation? It involves alterations in external form, including color, shape, size, etc. For example, albino ascospores in Neurospora, kernel color in corn, curly wings in Drosophila, and dwarfism in pea. Biochemical mutations are identified by a deficiency so that the defect can be overcome by supplying the nutrient or any other chemical compound for which the mutant is deficient. For bacteria, biochemical mutants need to be grown on a media supplemented with a specific nutrient. Such mutants are called auxotropes. Mutations based on cytology are chromosomal mutation, gene or point mutation. Chromosomal mutation is of two types. Number 1, change in chromosome number, and number 2, chromosome aberration. Change in chromosome number. In this type of mutation, an entire chromosome may either be lost or get duplicated, resulting in an aneuploidy. If a chromosome is gained, it will result in the formation of one chromosome, which will have 3 homologues and will be referred as the trisomic. The loss of one chromosome will result in one chromosome having only a single homologous, and the organism can be referred as a monosomic organism. Sometimes it may be the loss or gain of a complete pair of chromosomes, resulting in the formation of euploidy. Number 2, chromosome aberration. Chromosome aberration or extensive damage in the DNA, which may be due to breakage of chromosomes or due to transposition of the transposon. Such mutations result in major changes in genetic information and are caused due to some unusual events, such as in the survivors of atomic bombs exploded in Japan in Second World War. Now, gene or point mutation. Point mutation consists of single change in the nucleotide sequence. If the change is a replacement of some kind, then a new codon is created. In many cases, this new codon upon translation results in a new amino acid. When base change results in new amino acids, new proteins appear. These new proteins can alter the morphology or physiology of the organism and result in phenotypic novelty or lethality. Point mutation that occur within the protein coding region of a gene may be classified into 3 kinds, depending upon what the erroneous codons codes for. Number 1, silent mutation. Silent mutation are mutations that do not result in a change to the amino acid sequence of a protein. They may occur in a region that does not code for a protein, or they may occur within a codon in a manner that does not alter the final amino acid sequence. Number 2, missense mutation, where a single nucleotide is changed to cause substitution of a different amino acid. This will cause the resulting protein non-functional. In other words, missense mutations code for a different amino acid. Number 3, nonsense mutation, which code for a stop and can truncate the protein. Now, mutations based on molecular mechanisms are base substitution and frame shift mutation. Base substitution, a type of mutation involving replacement or substitution of a single nucleotide base with another base. It is of 2 types, transition mutation and transversion mutation. Now, what is transition mutation and transversion mutation? Transitions are routinely produced by base analogs. 2 of the most widely used base analogs are the pyrimidine analogs 5-bromouracil and the purine analogs 2-aminopurine. The mutagenic mechanism of the 2 are similar. The 5-bromouracil is incorporated into DNA in place of thymine. It acts just like thymine in DNA replication. Bromine atom cause 5-bromouracil to tautomerize more readily than thymine does. Thus, 5-bromouracil goes from the keto form to the enol form more readily than thymine. Transitions frequently result when the enol form of 5-bromouracil pairs with guanine. The 2-aminopurine is mutagenic by virtue of the fact that it can pair with cytosine. Thus, at times, it replaces adenine and at other times guanine. It promotes transition mutation. Thus, transition means if the replacement of one base pair maintains the same purine-pyrimidine relationship. For example, adenine-thymine is replaced by guanine-cytosine and guanine-cytosine by adenine-thymine. In both examples, a purine-pyrimidine combination is replaced by a purine-pyrimidine combination, or more specifically, a purine replaces another purine. The mutation is referred to as transition mutation. Now, transversion. Exposure to mutagens results in loss of bases. These sites where this happens are referred to as apurinic or apyrimidinic sites. If the AP site is not repaired, any of the 4 DNA bases would be inserted into the new strand opposite the gap. When a purine replaces a pyrimidine or vice versa, it is referred to as transversion mutation. For example, an adenine-thymine base pair can be converted to a thymine-adenine base pair. Frame shift mutation. Addition or deletion of a base are potentially the most devastating in their effects on the cell or organism because they change the reading frame of a gene from the site of mutation onwards. A frame shift mutation causes 2 problems. First, all the codons from the frame shift will be different and thus yield a useless protein. Second, stop signal information will be misread. One of the new codons may be a nonsense codon, which causes translation to stop prematurely. Or, if the translation apparatus reaches the original nonsense codon, it is no longer recognized as such because it is in a different reading frame and therefore the translation process continues beyond the end of the gene. Mutations based on survival or lethal mutation. It involves genotypic changes leading to death of an individual. These are perhaps the easiest to score for a study of mutation frequency. Mutations based on effect on function. Number 1, loss of function mutation. Wild type alleles typically encode a product necessary for a specific biological function. If a mutation occurs in that allele, the function for which it encodes is also lost. The general term for these mutations is loss of function mutations. The degree to which the function is lost can vary. If the function is entirely lost, the mutation is called a null mutation. It is also possible that some function may remain but not at the level of the wild type allele. These are called leaky mutations. Number 2, gain of function mutation. It leads to change in the gene product and it gains a new and abnormal function. These mutations usually have dominant phenotypes. This type of mutation is often called a neomorphic mutation. Number 3, dominant negative mutation. This type of mutation leads to an altered gene product that acts antagonistically to the wild type allele. These mutations usually result in an altered molecular function and are characterized by a dominant or semi-dominant phenotype. It is also called endomorphic mutation. Mutations based on fitness are beneficial mutations, harmful mutations. Now, what are beneficial mutations? Many mutations allow an organism to be more suitable to the survive in an unfriendly environment is called beneficial mutation. These mutations permit the development of a new characters while also give the adaptability to the surrounding. The induced mutation can be used for providing new characters. The application of such mutations have been more in plant science. It has been possible to provide an organism with useful characters such as pest resistance, stress resistance, or high yielding varieties. For example, it has been possible to develop the variety of penicillium which has higher yields of penicillin. Now, harmful mutations. A harmful mutation is a mutation that decreases the fitness of the organism. Special class of mutations are conditional mutation. Conditional mutations are those which allow the mutant phenotype to be expressed only under certain conditions called restrictive conditions. For example, high temperature. Under other or normal conditions called as permissive condition, the mutant expresses normal phenotype. This is all about mutation and its types. Now, to sum up, mutation is the source of genetic diversity and raw material for evolution. In this discussion, we have learned about classification, causes and mechanisms of mutation. Thank you very much.